I am from the Burnett Mary Regional Group. I'm the research manager there, and we are a natural resource management group, much like Desert Channels, who we heard from earlier. Our area is much smaller than Desert Channels, so it's from about 1770, just south of Gladstone, just in behind the Suncoast hinterland, but we also cover Fraser Island and out past Mundubbera. So our area is really rich in threatened species. We have several endemics like the Mary River cod, Mary River turtle, and a few frogs. We have numerous species which are limited in their distribution, like white-throated snapping turtle, Nanga spiny skink, and the Oakview leaf-tailed gecko. And then we also have many species which have direct impacts from degraded habitats and management practices, such as our koalas, our glossy blacks, quolls. So that we have a strong focus on restoration of these habitats with our Indigenous partnerships. So working in Indigenous knowledge into all of our projects and whatnot. And where there isn't a current solution that's working, trying to build on innovation to get us to a new spot, whether that's positive or negative. Hopefully always positive though. So this is part of the team. As you can see, we're a wild bunch. So our CEO, Tom Espinoza, down on the bottom right-hand side, he's the brains of a lot of the innovation. He's got lots of ideas, and we are sort of the delivery system of those ideas. And we also have two traditional owners in our group. So we've got Des Purcell, who's a proud Tarrabilling Bunda man, and Conway Burns, who's a proud Bachelor man. And so they help us to reach out and engage with the TOs to ensure that there's traditional ecological knowledge in all of our projects that we research. So our threatened species toolkit mostly, is related to three main types of tech. And then if I get a chance, I'll talk about one of our innovation, which is the simulated rainfall. But cameras are used across pretty much all of our projects. We largely use them with acoustic sensors, which we primarily focus on for riparian health indices, as well as for birds. And then we've got our eDNA for species detection, both species of interest, such as wanting to know about the habitat of black-breasted button quails, which are a threatened button quail species in our area, as well as understanding the distribution and presence of threats. So such as for a macadamia nut species, the bull-burr nut, we have two main threats that we're interested in looking at, which is pigs and a disease called Phytophthora. So we're able to use eDNA to monitor both the species and the threats. But what's most important is none of these three aspects are done in isolation. They're all used with something else. So we don't just do eDNA. It's usually paired with cameras and cameras with acoustics. So there's always more than one way of monitoring what we're monitoring. So our cameras, as I said, are used on pretty much all of our projects. We can have just basic camera information is that you can have two different types of sort of images that come out. You can have motion activated images, which means that something walks across, takes a photo, and Bob's your uncle, you've got the image, and then you've got time-lapse image. So things that don't move fast enough to trigger the camera usually need time-lapse image. But for motion sensor image, where something is triggered in front of the camera, we can put those through a sort of AI sorting software. There's heaps of them out there, and that can sort of trigger and sort out where you've got an animal versus where you've got like a leaf hanging in front of the camera, setting it off. So that allows us to sort of do real quick sorting and then do the species ID from that, instead of spending, 3 days trying to sort through 60,000 images. We're able to just sort through the ones that are actually positive triggers. It's also good for cryptic and nocturnal species, which are difficult to survey for in the field. There's a few species that are really cunning and know when there's a human about, and so they will avoid it like the plague. But it allows us to do that from our office and still get the information of their presence there. And it can also be used for spatial and population analysis. So we can set up a grid and know where an individual is moving, where there's habitat that they're liking, and what those habitat features are. So we can do a lot more and expand on just the image itself. But as I said, you've got your two different types of settings. And so for one of our key threatened species that we're interested in is the Mare River turtle and the white-throated snapping turtles. They are slow-moving, cold-blooded things, so they need a time lapse in order to get seen on cameras, which means that you have loads and loads and loads of empty images that don't have a turtle in them, or don't have a fox in them, or whatever it is, but don't have any animal in them because you've had to take it every 2 minutes for 6 hours overnight. So that then leads to high storage needs. We've just got terabytes and terabytes of data. And the AI filtering that we use to sort where there's an animal and where there's not is not great at this time lapse sort of setup. So we have a photo there of a koala. We weren't aiming for a koala, but they have just been picked up when we're out there surveying for other things. So they're still really good at picking up those really cryptid species. And our AI has worked really well at picking up our images. So this is what looks to be a plain old landscape, but it actually has two black-breasted button quails in it. And if you have really keen eyes, you can see where they are, because it's glaringly obvious to the layperson. But I wouldn't have picked this up if I hadn't been for that AI sorting. And so those are the two black-breasted buttontails. And yeah, they just look like leaves to me. So I wouldn't have picked them up if it hadn't been for that sorting mechanism. And yet it doesn't pick up this white-throated snapping turtle coming up to nest, which seems more obvious to me, but it really struggled with it. So our acoustics aren't done alone either, so they're mostly paired with our cameras. They give us a really strong biodiversity signal. As Kieran was talking about earlier, they just blanket record. They're not biased with what they're listening for or anything, so they can also detect really cryptic species, so things that maybe don't want to come down in front of a camera, like your honey eaters, loads of different birds that it's not in their behavior to come down in front of a camera. And we can also use the soundscape that we get from that to infer a healthy or an unhealthy environment. But when they don't work is when you put them right next to, a road where you've got loads of road traffic, or right next to a running water. So that's where it gets sort of drowned out in the soundscape. But also at the moment, I am a threatened species ecologist. I'm not a software engineer, so I don't yet have the processing capacity to do all this fancy fandangled smart techie stuff with the bioacoustics. So we currently outsource that to people who know what they're doing with this. So that's one of our limitations rather than the software's limitations. So this is two examples of very conflicting sort of soundscapes. And while you'll think that the top bar, so at Mare River and Traviston Crossing, would be a good soundscape to be able to pick out loads of different sounds and loads of different birds and that, it's actually a really poor soundscape because you've got loads of black. So black is void, so it's empty space, it's empty noise, I guess. Whereas where you've got blue or where you've got green, that's sparse noise, and then red is your super dense noise. So you've got so much more black in the top because where the spot was right next to completely cleared, treeless dairy country, where you don't have the birds coming in to be able to sit down and make all their noise. Compare that to Amamore Creek, where you've got really rich, beautiful riparian zones, maybe a bit of weed, but they're still really thick riparian zones that are really dense and healthy, areas. So that's where you've got a really healthy land and soundscape, which you can see in the spectrograph. So environmental DNA, if I just take a step back, and I'm sure that the following presentations will sum it up a bit better, but everything has DNA in it. And when we go out into the environment, we shed some of our cells and some of our DNA into that environment. So we're able to test the soil and water and things that we interact with in the environment to see what has shed its DNA onto that environment. So we can test water to see what's come to it and to see what has, maybe touched it, to go drinking, or what has been swimming in it, all that sort of stuff. So we can look at a huge amount of biodiversity because loads of the animal kingdom has had its genetic knowledge sort of put on. It's been identified as their biodiversity genomes have been understood. So we can use that for early detections of things like those diseases, threats like pigs or carp that might come into our waterway. Again, it can help with cryptic species. And we've got two options when we're looking for eDNA. We can say, here's a sample, can you tell us the whole biodiversity that is in that sample, whether it's water or soil or air? Or we can look at a specific species and say, I'm only interested in this one or these few species. Can you tell me if they are present or not? So it's really wide and varying in its ability. It's also a very new technology in terms of application for management. So it doesn't have perfect results yet. You've obviously got, you can still miss species that are present, and you can also get those false positives. And there's a couple of areas where you just need to know how the species behavior to know when timing can be key. So this is our whole biodiversity assessment of our region, but we can also look at specific species. So the white-throated snapping turtle is a critically endangered turtle species. She was on the front of my slides, and you can see that she's got really dense sort of detections in the Mary catchment in the bottom, and then in the Burnett catchment sort of in the middle. And that's where we do two very extensive nest protection programs for them. And unsurprisingly, we have feral pigs spread throughout our region. They're just everywhere. But if we go back to our white-throated snapping turtle, one thing that they've missed is that very top creek line is the Baffle catchment. And that is where we have recently discovered a new population. So they were never known to be there before. They were only thought to be in the Mary, the Burnett, and then the Fitzroy catchment. But we've discovered a new population in the baffle, and obviously there were no detections when we were doing this eDNA sampling. And as well, our foxes. So Blind Freddy can tell you that there's more than those 5 foxes. You don't even have to know our region to know that there's got to be more foxes than that in our region. And this comes back to the timing. So if we look at when we sampled, in January, we sampled when there was heaps of water around. So foxes, unlike pigs, don't necessarily need to come to a water. So pigs will wallow and spend a lot of time in the water or wet areas, whereas foxes don't really need that. And then our second sampling was in July, and July is when a fox will be tucked up in its den with its kids. So that's probably why we didn't get very strong signals for foxes, just purely on timing. And then if we go to an example of our innovation, so this was when we were trying to prepare and get ahead of the times, in that it was meant to be coming into dry period, and we wanted to see if we could sort of trick turtles into laying nests if their rain dependent signals were going to be delayed or not at all coming, like we would see if we were going into a drought. So we set up an irrigation system that allowed us to, remotely turn on and off irrigation, access some real-time tracking of the moisture levels in the soil. And there have been so this was a pilot study that then got adopted elsewhere, and there's some indications elsewhere that this sort of system can trigger turtle nesting events outside of a typical rain event. But yes, it does come with its costs. There was so many battery failures, data storage. Our area flooded multiple times repeatedly, and it ended up, rather than being a low rainfall year, we ended up going into 2 years of above average rainfall. So timing was an issue as well. So that's the system on the left-hand side, and then just 2 images on the right-hand side. So this is how much this river changed over 12 hours. So that's 6 o'clock in the morning, and that's that same area 6 o'clock at night. So it's a really dynamic system that we weren't able to, that we didn't fully understand. So for us, start small, pilot early, like we did with the irrigation. Even though we didn't prove that it worked because we had many issues, it's still gone on to have really good impacts elsewhere. Like with the bioacoustics, we need to research, sort of join with tech partners and researchers who know what they're talking about. Combine tools for reliability, and across all of our projects, we embed traditional ecological knowledge throughout.
Thank you, Stephanie.